All right, group, this is your last set of notes for this note packet. So by the time you get to the end of this set of notes, the note packet from Unit 2 should be completely filled out. So let's jump into it. Okay, so for the organelles, we've already talked about the basics for the cell membrane and the cell wall, but in this part of the notes, we're going to get into more details, really the structure, the function, how everything works, what the process is when things come in and out of a cell. So we're just going to get into a little bit more detail. So cell boundaries, these are going to be those things that maintain that this cell is its own entity while this cell is its own entity, and that though they can work together, they are still separate from each other. So the cell membrane is found in every single cell possible, from prokaryotes to eukaryotes, plants, animals. All cells have to have a cell membrane. The cell membrane is going to be flexible, and then its job is to pick and choose what can come into the cell and what can leave the cell. Its structure is made up of phospholipids, which is called a lipid bilayer. And that word bi, or that prefix bi, means two. So the lipid bilayer means that there's two layers of lipids. So they kind of dangle like this on top of each other if my fingers are representing the lipids. And then the phospho part is called the phosphohead. So that would be like the back of my hands. So they kind of set up like this. Cholesterol is another part of the cell boundary or of the cell membrane, and its job is to help with cell communication. Proteins it allows cells to link up with each other, but allows stuff to come in and out. And then carbohydrates is a major part of like what they're made out of. The cell wall is only found in plants, bacteria, algae, and fungi cells. So no animal cells have a cell wall. The cell wall's job, it's not there to regulate what can come in and out of a cell. The cell wall's job is to provide support and structure. So it's very strong, especially because the organisms that have cell walls, they live in the harshest of harsh conditions, and so sometimes they need that extra support. Or like in plants, they have to stand upright. Their roots or their stems have to go upright. So they need that cell wall to make sure that they can stand upright in the best of conditions and the worst of conditions. In the cell wall is made up of cellulose if it's found in plants, which is it's a type of sugar. Or it can also be made up of chitin, which is what the cell wall of a fungus or fungi and protists are. So fungi would be like mushrooms. The fluid mosaic model, this is the term that we use to describe how the cell membrane works. So the fluid part of it means that there's movement with it, that it's flexible, that it is not just this rigid structure there that you can poke it and it'll kind of poke in like a balloon. The mosaic part means that everything kind of fits together perfectly, just like with the mosaic artwork where you have the little pieces of glass or the pieces of rocks, and they've been cut to make a picture. Same thing kind of here, where you have all of these phospholipids and the proteins, and everything just kind of fits together perfectly. So the whole thing, the fluid mosaic model, means that everything fits together perfectly, but they all move together basically as one. Phospholipids, as mentioned, have two parts. You have the phosphoheads, which are these parts up here, the little yellow circles, and the polar heads. So the phosphoheads, they like water. They can touch water. They can come into contact with water because they're polar. And there's this thing in chemistry and in biology. It's called like dissolves like. And this just means that the polar heads, because water is another polar molecule, which doesn't mean a whole lot to you right now because you haven't had a chemistry background yet. But essentially what it means is that the atoms that make up water have some charge to them. Well, the phosphoheads also have a similar charge to them. And so because of that, they like each other. They can come into contact with each other. The nonpolar or the lipid tails. So lipids are fats and oils. And if you've ever done any amount of cooking, or you've ever tried to mix oil and water, you know that they don't like each other. They hate each other, and they will try to stay away from each other as much as possible. So the same thing is true here with the lipid bilayer, where you have these lipid nonpolar tails and they hate water. So that's why they kind of face each other. If these are my lipid tails, they face each other because they don't want to come into contact with any water. So if water does come close to this cell, the cell can essentially like wrap around itself to protect it from the water. So the membrane's function overall is that its job is to maintain homeostasis. So what homeostasis is it's the process of maintaining the cell's environment. So we have a lot going on inside of our cells, just like with our bodies, we have a lot going on. Cells have to maintain a correct like acidity or pH level. They have to maintain a correct salinity, how much salt they have. Cells have to maintain, if possible, a correct temperature, and there has to be ways that if any of those things get off, they're off track, the cell has to be able to bring them back on track. So homeostasis is what all of this is about, is maintaining life in a way that is conducive for the cell or for the organism as a whole. Selective permeability, this is, and we've talked about this before we took our vocab quiz. If something is selective, that means it's choosy, it's picky. So like colleges can be selective. The early college program can be selective. We can pick and choose like for colleges, the students that they want. They can reject students that they don't want. And then permeate or permeability, that means to like come in. So permeate means to like go through. So selective permeability means that the cell membrane's another function of it or another job of it is to allow some stuff in and allow some stuff out, but they don't always have to let things in, let things out. Like it chooses based on the homeostasis of the cell. Okay, cell transport. There's two major types of cell transport, excuse me. You've got passive transport. And the way I remember passive transport is if somebody's passive, they're just like, okay, not a lot of energy there. So passive transport is the same. This is movement that does not require any energy. You don't have to or the cell does not have to exert energy in order to get this movement or this transport to happen. And there's three separate types of passive transport, and we'll go into all three of these, but they are diffusion, facilitated diffusion, and osmosis. Active transport, just like if somebody is active, they have a lot of energy. They have to exert that energy. So active transport means it's moving stuff in and out of the cell, but it has to use energy. It has to use ATP to do that. So three types of active transport are your transport proteins. Endocytosis, which means endo means into, cyto means cell. So this is the process of bringing things into the cell. And then there's two subtypes for that. And then exocytosis, which means taking stuff out of the cell to exit the cell. So to go into more details about diffusion just simply means it's the movement of particles. So in your notes, you might underline or circle that word particles. So diffusion is specific to stuff that is not water. Typically, we'd say it's stuff that's like solid, but it's really anything that's not water or water-based. So diffusion is the random movement of particles of stuff from an area of high concentration to an area of low concentration. So how I think of this is if, let's say, for example, in my classroom, so think about the size of my classroom. Let's say we tried to fit all 450, I think that's how many freshmen there are. But let's say we tried to fit all the freshmen in my classroom. So everybody is jam-packed in here. And then I open the door. Because of the amount of concentration of students, once that door opens, it is like everybody's just going to flow out. Whether you want to or not, you are probably going to get caught up in that flow of students, and you will then exit my room. Okay. So diffusion means that you start with high concentration. So that's what we're looking at here. And then as soon as the opportunity presents itself, it's going to move to an area of low concentration where there's more room, there's more space. Eventually with diffusion, everything will disperse evenly. So let's say again that this is my classroom and we do start with 400 students or 450 students. Once it's about 50-50, so everything kind of looks the same. So we have 50% on one side, 50% on the other. Then we go into this process that is known as equilibrium. And this is where students can come in and out equally. So as long as the containers are the same size. So equilibrium, uniform concentrations, and this does not mean that particles stop moving. So that's also something to remember. This just means that particles can move, uh, they'll move in equal amounts. So there's still movement. So you might add these to your notes. There's still movement, but in equal amounts. So I would make sure I add that to my notes. All right. So in diffusion here is an example of that. If you drop some ink or some food coloring into a beaker, especially a warm beaker, the ink particles really start off concentrated. But if you let it sit there long enough, eventually the ink or the dye will separate and they will become equally distanced from each other, which will result in the beaker looking all one color. So those particles are still there and they're still different than the water particles. They've just spread out. And so then the color slightly changes. And that's also why the dye looks really dark right here, but then looks lighter, is because the concentration's really condensed and then they separate out. Okay, facilitated diffusion is very similar to what we just talked about with like normal diffusion. The difference is with facilitated diffusion, we have to use proteins to make this happen. And that's because the particles, they're too big to fit through the cell membrane. So in some cases, they can just kind of wiggle their way through the cell membrane. If the particle's too big, it has to use a protein, also known as a channel protein, in order to get through. So same principle. We start with a high concentration on one side, low concentration on the other, and the with facilitated diffusion, the particles will have to go through that protein to get to the other side. So still diffusion process, high concentration to low concentration, will go until equilibrium is met, and then the particles will move equally, but it has to have that helper. And I think I mentioned during the vocabulary before the vocab quiz, facilitated diffusion would be like the door in the classroom. We're too big to fit through the wall. We will not make it through the wall. But if we open that door, we can use that as a facilitated diffusion method or as our channel protein so that we can get out. Osmosis is the third